Hey guys, what's going on? It's T-Carp for the next reading of The Truth Denied. And we're reading my dad's book, again, The Truth Denied. It's his rendition of the Sasquatch DNA Study. Guys, we've read the introduction and chapter 1. Today we're going to be reading chapter 2 and getting into which is called The Beginning. So getting into how it happened, what's going on, and all that. So if you haven't checked out the other videos, go ahead and check those out too, so you kind of understand what's going on. But guys, let's get right into this reading. Chapter 2, In the Beginning. In early 2009, the TV show Destination Truth, starring Josh Gates, brought a hair sample back from the Bhutan. In May of 2009, the producers of the show chose Dr. Melba Ketchum of DNA Diagnostics to perform DNA analysis. Dr. Ketchum did not believe that the creatures known as Bigfoot existed, but she agreed to run the analysis anyway. What she found astonished her. The mitochondrial mtDNA tested human, but the hair was not human morphologically. When Dr. Ketchum compared the sequence to GenBank, GenBank is a database of millions of DNA profiles, both animal and human, she could not find a match. Below is the transcript from an interview of Dr. Ketchum by Josh Gates that aired November 5th of 2009. Dr. Ketchum: I didn't think we would have anything to talk about here, to be honest. I was just going to rule out Yeti and be done with it. I submitted the sequence that we obtained from this hair sample to a large international database that scientists use to deposit their sequence data. Well, at first, I was very skeptical because we've had these things come out of our lab in the past and they never panned out to be anything interesting. However, this sample did test very clearly on the human panel of markers. That makes it a primate and makes it a large primate, Gates. And how are we sure this isn't just human contamination or just DNA from human hair? Dr. Ketchum: the hair visually is not human. It's coarser than a horsetail. Gates, voiceover: what she told me next seemed unbelievable. Dr. Ketchum: initially, searches indicate that it's an unknown sequence. There are literally millions of sequences in the database, and we're really shocked that it didn't match any of the species exactly in the database. Gates: what would be required in order for us to say, from a DNA standpoint, that Yeti is a real animal? Dr. Ketchum says, if we're going to prove that there potentially is a new species, with this first hair sample, we really need more hair samples like it. And once you establish there is a group of animals, that will go a long way towards proving that there is a new species indeed. The Josh Gates hair sample had Dr. Ketchum curious, along with now being open-minded about the possibility of Bigfoot being a real, living, breathing creature. In an interview, Dr. Ketchum explains how these results changed her mind. Quote, I have an insatiable curiosity and love a good mystery, so it sounded like something I would enjoy investigating. I really didn't believe that they existed and thought I would just disprove it and go along my merry way. I was totally oblivious to what was headed my way with this. It never occurred to me how difficult, quote-unquote, proof would be and how long it would take. We had tested, quote-unquote, alleged Sasquatch samples since 1995 for species ID, but they had only tested as common animals up until early 2009. Even then, I didn't trust my results, as there wasn't enough DNA to continue with those samples. So I just wasn't expecting this, nor did I think about the implications since I didn't believe in them. In 2009, there was a convergence of circumstances that brought Dr. Ketchum, David Pilates, Richard Substad, Adrian Erickson, Wally Pearson, Bob Schmalzbach, and others together. David Pilates. Spring-summer of 2009, Pilates had been unsuccessfully searching for a lab to test possible Sasquatch hair for DNA. David would inform the labs the subject of the testing, causing them to immediately decline. I find it ironic that these labs would actually turn down revenue, which tells one of the stigmas attached to the word Bigfoot. David finally found Dr. Ketchum, who was open to testing his hair samples. The following is from the NABS website. David is explaining in his words the quote-unquote start of the genome project. NABS has had the funding to make a sustained effort to the DNA issue. The efforts started by locating the optimum location to test the specimens. NABS interviewed several scientists. The interest level and competency ran the gamut. One person stood out amongst all others interviewed, Dr. Melba Ketchum of DNA Diagnostics. We explained what our mission statement was and our sincerity to keep this at a highly scientific level. NABS started this DNA study, and we will be there when it completes. Dr. Ketchum stated she was interested in the NABS sent her our first specimen hair found on the Hoopa Reservation in Northern California. As part of the hair recovery and identification process, Dr. Ketchum employed the services of hair and fiber expert, and she had previously known through his expert courtroom testimony. This individual works for a prominent institution who allowed him to work with our project and volunteer his time. This person reviews all hair samples and compares them to all known hair and fiber and ensures the samples meet the criteria for Bigfoot/Sasquatch/hairy man. He photographs the hair and then passes it on to Dr. Ketchum for DNA testing. If the specimen doesn't pass the hair and fiber testing phase, it does not move on to DNA testing. It should be stated that Bigfoot, Sasquatch/hairy man has a distinct physical features that does not match any known hair or fiber. This person reviews all hair samples and compares them to all known hair and fiber and ensures that the sample meets the criteria for Bigfoot, Sasquatch, or hairy man. He photographs the hair and then passes it on to Dr. Ketchum. So I kind of want to interject here. So they didn't just test every single hair. They looked at it and made sure it met the qualifications, because you can look at it in the microscope and tell, like, if it's possum hair, if it's dog hair. You're not going to spend the money to test the DNA test a dog hair. Like, you kind of know. It has to fit the physical specifications, which is very wild and interesting in itself as well. Dr. Ketchum had been testing samples many years before David/the NABS approached her. The NABS samples were the first with results pointing to the existence of the species. However, the project was not born until the Destination Truth aired. This provided the interest that would be needed to get the funding and the volume of samples to start the project. Richard Substad. Substad joined the project in January of 2010. He had become interested in Sasquatch Bigfoot after a family reunion in the summer of 2009. He was challenged by a cousin to take a closer look into the existence of Bigfoot. When Substad investigated the Bigfoot phenomenon, he found enough evidence to further pique his interest. Substad contacted Adrian Erickson, Robert Schmalzbach, David Pilates, Shannon Silva, and Dr. Melba Ketchum in January of 2010. Adrian Erickson of the Erickson Project. Erickson joined the study in March/April of 2010. The Erickson project had been underway since the 2006-07. Erickson had encountered issues with the labs that had been testing the samples collected from his Kentucky property. Todd Distol, an anthropologist from NYU, tested a sample of blood and flesh. Dr. Ketchum gives an accurate summation of the results along with Distol's reactions to the results. Quote, Todd Distol is a perfect example of the bias and intolerance this study has faced from the beginning. Distol was involved in a 2009 in testing a flesh blood sample from the Erickson project from a red female Sasquatch, which was later included in the Sasquatch Genome Project study. Distol analyzed only the mitochondrial DNA, or mtDNA, from the sample 37 in the SGP study, and found it had modern human mitochondrial DNA in the H. haplop group, or HAPLOGROUP group. Okay? Distol assumed that this was represented human contamination. Okay? And so threw the sample away, which outraged the leaders of the Erickson project. The mitochondrial DNA result was confirmed by Paleos Labs and DNA Diagnostics, the two other labs which studied the sample. But at DNA Diagnostics, the sample's nuclear DNA was tested, which confirmed it was from a female and expressed the MCR1 gene for red hair color. No red-headed females were involved in the Erickson project. When the sample's nuclear DNA was run as a human genome, was run on a human genome SNP array test at USC, it ran at only 80%, well below the human threshold of 95%. Even the study's control degraded human blood sample, which had been purposely left to decay, ran at 97%, indicating the low percentage on sample 37 was caused by genetic differentiation and not by degradation. So let me try to kind of explain that in terms we can kind of all understand for just a second. What we're saying, what they said there is, there was a test of this sample by a red-headed Bigfoot, okay, red-headed female Bigfoot. What they tested is the mitochondrial DNA, which is the DNA, or which is the mitochondria, the outside part of the cell. Okay, the outside part of the cell usually represents the mother's DNA. So that was human, but when they tested the nuclear DNA, the DNA of the father, that was 80%. So like he was saying, well below the threshold of human DNA, and they ruled out like degradation or like decay by having an absolute kind of like a what they call a baseline sample and left it to decay, and it was 90% human. So what they're saying is like there's no way it was just decay. So that's kind of what they're saying, is the mitochondrial DNA was that of human, which again with this study we'll talk about later. The nuclear DNA was not. So very, very, very interesting. Very, very interesting stuff. And we're getting into DNA stuff. So anyway, the next is an excerpt from the email between Todd Distol and Dr. Leela Hajika. Leela, it's 100% matched to multiple humans' mitochondrial DNA genomes, including haplotype HEU7445412. Todd R. Distol, Professor, Center for the Study of Human Organs, Department of Anthropology, New York University. The Paleos lab results. When the DNA sequence was compared to a genetic database, NCBI BLAST, Basic Local Alignment Search Tool, nucleotide database, it identified the DNA as belonged to Homo sapiens. An independent alignment of these sequences to the universal human mtDNA reference sequence called the Revised Cambridge Reference Sequence, or RCRS, was done. The alignment indicated a 100% match to Homo sapiens mitochondrial DNA. Multiple mutations are present in other higher primates within the region. The DNA extracted from the swab was also subjected to a universal DNA detection test for the Site B region of the mitochondrial DNA. No results were obtained. The primers used for the Site B region will be direct birds, mammals, reptiles, and fish, but are not human specific. Based on the above findings, it is my opinion that the blood stain is of human origin. Technical manager Stefan Fatipiro, or that's from that. More documentation of what happened between the Erickson project and Todd Distol was posted by the official spokesperson for the Sasquatch Genome Project, Robin Lynn, via Facebook. All right. So, again, just kind of trying to tell you what they're doing. They're testing these mitochondrial DNA, and it's coming out human. It's just coming out human. It's coming out human. They're not testing that nuclear DNA, which that's the big thing. So again, this is via Facebook. This needs to be said. To clarify, I, Dr. Ketchum, do not know Todd Distol. When Adrian Erickson came to our project, he told me that he had sent an aliquot of red Sasquatch's blood to Distol for testing. AE, or Adrian Erickson, has results and corresponds as proof. He was extremely upset because Distol had destroyed the sample. Distol got human mitochondrial DNA sequence when he tested the sample and didn't believe that it came from a Sasquatch. He told the PhD on Erickson's staff that somebody must have hoaxed it. He said this in spite of the fact that the study was well monitored by Dennis Foll, who is extremely credible, was in control of the food trap start to finish. If it was hoaxed, our genetic testing showed the suspect would be a red-headed female, which obviously Dennis is not. In addition, the whole genome SNP testing excluded this individual, the red-headed female, as being 100% human, which results close to 20% below the human threshold. So what they're talking about is when the nuclear DNA testing was done, again, it was 80%, which is well below what a human nuclear DNA is. So it's not human. So it's a really hard thing to kind of grasp. After Distol destroyed the DNA, Mr. Erickson's staff had requested the remaining sample be returned. Another aliquot that had been retained by Erickson was sent to Paleo Labs in Canada. They also obtained human results on the mitochondrial DNA. Finally, Adrian Erickson sent the remainder of the DNA to our study and had Paleo Labs send what they had left also. Our lab also obtained human mitochondrial sequence. These are all independent tests using different laboratories. When we then begin testing the sample using several platforms to obtain nuclear DNA sequences and profiles. This sample, like all other samples in the study, yielded human sequences as well as unknown sequences. That's the key in my opinion. Sorry, I need to keep reading. First results. In these early days of the study, the team dynamic was extremely fluid. The results were being analyzed with new revelations occurring almost daily. The study started with 3 samples. The 2 most important samples for the study at this early date were called Sample 1 and Sample 2. Sample 1 was a purported Bigfoot toenail found on the Bigfoot habituation site in northwestern Arizona. Sample 2 was a blood and tissue sample collected in Kentucky. Analysis of these 2 samples were the aha moment for the study. On his own and separate from the Sasquatch Genome Project, Richard Substad did statistical work. In June of 2011, he published a blog article detailing conclusions based on his separate analysis. The 2 purported Sasquatch specimens are closely related to one another. Sample 1 has 2 polymorphisms, while Sample 2 has 3 polymorphisms. Various mitochondrial DNA sequences are shown along the x-axis as examples. This is the Cambridge reference sequence. Most likely, these 2 specimens, widely separated within North America, share the same mitochondrial Eve, who probably existed somewhere between 20,000 and 30,000 years ago, based on the number of mutations or polymorphisms presented in the mitochondrial DNA genomes. This mitochondrial Eve was most likely European in origin, belonging to some version of haplogroup H. This haplogroup occupied the so-called Franco-Cantabrian glacial refuge, covering the late Wisconsin glacial period when most of northern Europe was covered by glaciers. This is around the middle of the same epoch when the continent of Beringia existed, creating a 1,000-mile wide Asia-North America land bridge. The odds of a random hoax or misidentification for either Sample 1 or Sample 2 are 2% or less statistically. The 2 purported Sasquatch samples were not physically connected whatsoever. Rather, they were obtained from 2 disparate groups and individuals from 2 widely separated geographic locations within the continental United States during 2009. Thus, the odds of either being a hoax or misidentification are less than 2%. Let's say 3% conservatively. No current GenBank Homo sapien genomes perfectly match either Sample 1 or Sample 2. What that means is this GenBank, which is a bank of millions of DNA things, didn't perfectly match either Sample 1 or Sample 2. So I just want to, again, get that clear. Several ancient ancestry Europeans from haplogroup H match Sample 1 within 3 pairs and Sample 2 within 4 pairs. This translates to an approximately 99.98% mitochondrial DNA match of the entire genome for both purported Sasquatch specimens. The source of this, again, can be found the Sasquatch DNA Research, June 2011 update by Richard Substad. From a YouTube interview, and now this is from a YouTube interview of Richard Substad. Quote, much to his surprise, once again, the first 2 samples submitted by 2 totally independent and disparate researchers from the habituation sites in 2 widely separated states or provinces turned out to be initially related to one another, DNA-wise, making the statistical probability of the 2 independent hoaxes or misidentifications somewhere in the 2 to 3% range. Accordingly, Richard's statistical conclusion from only having analyzed the mitochondrial or the prehistoric maternal origins, remember the mitochondrial DNA, sequencing of these first 2 samples alone is that there is about a 97 to 98% chance certainty that the Sasquatch or Sasquatch indeed existed right outside your own back door, so to speak. I have placed in bold more of the findings and quotes and stuff like that. Dr. Ketchum decided testing had to be complete, especially on the nuclear DNA. The testing of the nuclear DNA and results would place this study into high gear. And with that was chapter 2 in the beginning. So in that, we covered mitochondrial DNA, what they're seeking for, the nuclear DNA, how they couldn't be hoaxes, what the percentage chance was, how the mitochondrial DNA linked up to human had a natural Eve or the same Eve, and these things came from two different parts of the United States. So the chances, just trying to tell you guys the chances of this is extremely low of being a hoax. Anyway, guys, thank you so much for watching. Again, this is T Carp signing out. Thanks for watching The Truth Denied. Stay tuned for another reading here soon. And guys, love you. God bless.